response to light. It only takes one photon for rods for them to be active. What happens is the light hits the rhodopsin, and the energy of the light, the photon, changes the shape and the interaction between retinal and auxin. And that causes the transducin to release one of its subunits, which interacts with phosphodiesterase. So that is now activated. And phosphodiesterase in most of our other cells breaks down cyclic AMP. In our rods and cones, phosphodiesterase breaks down cyclic GMP. And so,
Say that again.
and usually phosphodiesterase functions to break down cyclic AMP, but in the case of the rods, it's breaking down cyclic GMP. With less cyclic GMP, there is no, or less cyclic GMP to bind to our sodium channels. So our sodium channels close. Sodium does not enter the cell, or at least less of it does. So now what's happening to the potential inside the cell? Is it going to become more positive or more negative? More negative, because our potassium channels are still open. They're not affected by the cyclic GMP. So we're not gaining the positive ions, but we're still losing the positive potassium ions. So the cell becomes more negative. It repolarizes or hyperpolarizes. Our minus 40 goes down to a minus 70. Potassium channels no longer receive a threshold. So potassium channels, what color were they, blue? Potassium channels now close. I'm sorry, calcium channels now close. And therefore glutamate is no longer released. Vg is voltage-gated. So as a result of the light energy affecting the rhodopsin, we stop the sodium from coming in the cell. We close the calcium channels. Synaptic vesicles with the neurotransmitter glutamate no longer bind to the presynaptic membrane. Glutamate is no longer released. Yes, sir.
Did you put X's through the GMP because
Because the phosphodiesterase is breaking it down. Yeah. So what result does this have then on the downstream neurons in our sensory pathway? This diagram here is partially correct. It says in the dark, photoreceptors are depolarized, calcium channels open, and the neurotransmitter is released. That's true. In the light, the photoreceptors are hyperpolarized, calcium channels are closed, and neurotransmitters are not released. This is partially correct, because it'll say glutamate causes an IPSP, an inhibitory postsynaptic potential. So when glutamate is not released, no IPSP is created and the cell depolarizes. It depends, it's true, but it depends on the cell. So I want to diagram that for you, okay? Yes?
Is there not supposed to be sodium in the rod part on the left?
Here?
Yeah,
Not when this is, so when this is happening,
right.
when it's in the light, yeah, this will not be there because it is hyperpolarized, okay? So, what I want to do now is draw this cell smaller, a little higher, and add the bipolar cell, okay? So, when you're done copying or taking a picture of it, I'll show you the bipolar cell on there.
Okay, so real quickly on those two, one says no neurotransmitter release, so that's
That would be
light.
in the light, exactly correct.
And then, but what about when it's, so does it start declining? Okay, so on the neurotransmitter that's released,
Uh-huh.
does it start declining, like if those channels start closing, or is that?
Well, this is in the dark,
Uh-huh,
so the reason the neurotransmitter is being released is because we're sending a creative potential
I get that.
down there, and the calcium ions are binding, releasing that. So the light, if it lowers the energy enough, will prevent the neurotransmitter from being released because the calcium channels aren't opening. I'm not quite sure I'm understanding your question.
So it's either they release them or they don't, that's it, right? So,
Yeah, I mean, depending on, it'll flow down and then eventually stop depending on how long the light stimulus lasts. So a stronger light stimulus means more photons,
oh,
so
okay,
it might
that's what I mean.
last longer. Yeah.
Are opsin and rhodopsin interchangeable?
Rhodopsin is the form of opsin in the rods.
Okay,
And then we just talk about opsin S and opsin L and opsin M.
so like for test purposes, if you just have opsin, you know.
Yeah, okay, all right. Okay, can I erase this now? Okay, so what I wanna do, in the next to the last diagram I'm gonna go over with you, the last one's gonna be the pathway, so we're almost done here.
Oh, I'm so confused right here.
I was just about to say, isn't this clearer since we've actually already talked about G proteins and
Yeah, it is.
we've done it today for the first time?
Well, that part is helpful. Thanks for
All right.
making sense.
So here is the end
Check that out.
of a rod, okay? And this will be in the dark. And here's the same rod in the light. So in the dark, we are releasing neurotransmitter, right? Glutamate. That's what we just went through. So we have our calcium channels are open, a graded potential has been coming down. All right, so our dark current. Now, the bipolar cell is capable now of creating an action potential. I'm going to draw another bipolar cell over here. Here's why I said this was partially correct. Glutamate. It has two different kinds of receptors, right? Ionotropic and metabotropic. So this cell on this side, we're going to give it an ionotropic receptor. For sodium. So in the dark, if glutamate is released and it binds to an ionotropic receptor, is that going to create an EPSP or an IPSP? An EPSP. You guys are picking this stuff up. All right? So this creates an EPSP because sodium ions will enter the cell. Okay? Depolarizing the bipolar neuron. All right? Now, this particular bipolar cell, instead of having a ionotropic receptor, has a metabotropic receptor. Which functions to decrease cyclic AMP. So when the same neurotransmitter glutamate binds to this cell, are we going to make an IPSP or an EPSP? An IPSP. So the effect on the bipolar cell in the dark depends on what kind of receptor it has for glutamate. Some cells will be activated in the dark and some cells will not be activated in the dark. Now when we look at the effect of light, on those same cells, and we have an ionotropic receptor here, and a metabotropic receptor here, we have no glutamate because of the light effect, right? And the light. So now the opposite is seen. We don't open the ionotropic receptor, and so the inside of the cell is less likely to depolarize, less likely to release a neurotransmitter. This side of the cell is going to keep its cyclic AMP, so cyclic AMP is going to increase, and it's more likely to have other channels open and form EPSPs. So the exact opposite occurs. And it just depends on how many of these are activated. So if you're seeing an image, depending on where that light image is formed, and you're talking about the difference between edges and colors, and whether there's something moving across the vision or not, all of those are taken into effect by those horizontal cells and amacrine cells. So let's say that, and this is the end of required knowledge, all right? Let's just say that this cell, which is activated in the dark, and we said an EPSP was created, let's say that it releases glycine. Glycine is excitatory or inhibitory? Inhibitory. So even though we excited this cell, the effect downstream is inhibitory. Okay, so you can't always assume that just because one cell fires, that the result all the way down every neuron is going to be an excitatory effect, because it depends on the neurotransmitter released by that cell.
So you have an EPSP at one cell and then an IPSP at another.
That's kind of what we saw without going into the details of the basal nuclei. How excitation of one activity could cause inhibition of something else. Okay? And rather than knowing the direct sequences of all that, I just want you to know it can happen. all right, that we can get inhibition by exciting something that releases an inhibitory neurotransmitter. Or we can get excitation by inhibiting something that releases an inhibitory neurotransmitter.
So if those aren't depolarizing because there's not sodium coming in, how are you forming an EPSP?
Because this has more cyclic AMP, and the cyclic AMP can be opening other channels
Oh,
just
because
like
of
it
the
did, yeah. Okay, so it's just allowing it to respond, maybe to the horizontal cells or to other photoreceptors that are next to it. It gets really complicated when we look at all the neurons that are involved.
So what's kind of the difference between the dark and light, I mean, in terms of how we see things in the light? and-
All right, so when we are eventually, when we don't have all the images and we just block some images and see the ones that are already there. So eventually we are actually forming images. So when we see something, most of the reason we see something is because downstream neurons are activated. So most of our bipolar neurons would be activated in response to light. Okay, so that is this image that's shown here. Without the IPSP, then the bipolar cell is going to depolarize. So the illustration that's given here is for a cell that is metabotropic. Okay? So it would be this second one over here. So with light, the metabotropic receptor cell is going to be activated and start a sequence of current. These diagrams just look closer at the sodium channels and at the discs. Okay, and here's just, this right here is what I drew previously when I drew the membrane of one disc. So here would be the retinal, okay, with the cis formation changing to the trans, activating the transducin, and so here's the trans. This would be the transducin, and the transducin, which is really just G protein, activating the phosphodiesterase, changing cyclic GMP back to its non-cyclic form, and closing sodium channels. So it's just kind of what I drew on the board earlier. Okay, and these are just showing those illustrations. All right, so last concept that I want to cover, is the pathway, and I've drawn this already when we talked about the optic nerve, but I want to go over it again, just to go over it again, and to talk about lesions, what happens to vision if we were to cut the optic nerve or the optic tract. So here we have the retina, optic nerve, optic tract leading back to the thalamus, and then third order neurons leading back to the primary visual cortex. So this is actually what I'm going to diagram on the board right here. Okay, the same thing, just not in 3 dimensions. So may I erase the board? We have one last diagram to put up.